Hi everyone, and welcome to the second episode of our series Journey Through Lower Plants, Understanding Bryophytes. So in the last video we have discussed on the introduction of bryophytes, right? The general characters of bryophytes. So we'll have a short introduction. So bryophytes, as you can see here, these tiny little plants, they are known to grow in moist and shady places, okay, like on the rocks, on the tree barks. They are found to grow on the walls, and you will even find them in your garden as well. And these plants, they do not have any vascular tissues like xylem and phloem, and so these plants are land plants, but they require water for fertilization, okay. And this is the reason why bryophytes are also known as the amphibians of the plant kingdom. Now we have also discussed in the last video that bryophytes are divided into three groups, right? Liverworts, hornworts, and then mosses, right? So today we will study how each of these groups are classified, okay. So we'll study the classification of bryophytes today. If you haven't watched the first video, then I would request you to kindly watch the introduction, that is the general characters of bryophytes, and then come back to this classification. Alright, now we will start with the classification of bryophytes. Schimber and in 1879 gave Bryophyta a new rank of division. Okay, so before 1879, bryophytes were included along with algae, fungi, and then lichens. Okay, so bryophytes were grouped together along with algae, fungi, and then lichens. But Schimber in 1879, he gave Bryophyta a new rank of division. Okay, and then Bryophyta was divided into two classes. Class 1, that is Hepaticopsida, and class 2, that is Mossae. Hepaticopsida are also known as Liverworts, and Mossae are known as mosses. Now the classes Hepaticopsida and Mossae are again divided into three orders. The first one is known as marking shells, the second one is known as jungerminals, and then the last one is known as Anthocerotopsida. And then Mossae is also divided into three orders: specnils, entrails, and then bryals. Alright, but some of the bryologists considered Anthocerotopsida to be different from this class Hepaticopsida. Okay, the also argued that the order Anthocerotopsida should be given a new class. Okay, since it was different from Hepaticopsida, they argued that this should be different. This should have a separate class on its own. And later, the division Bryophyta was divided into three classes: Hepaticopsida, Anthocerotopsida, and then Mossae. All right. Now, Rothmeiler in 1951, and then Brosker 1957. Okay, they changed the names of the classes. Okay, so Hepaticopsida was changed into Hepaticopsida, Anthocerotopsida was changed to Anthocerotopsida, Mossae was changed into Bryopsida. Alright, so this is the classification of bryophytes. Okay, very simple, right? Now we will study each of these classes in detail. So we will first start with Hepaticopsida. Okay, the first class is Hepaticopsida, right? Hepaticopsida, they are also known as liverworts. Okay, so the name Hepaticopsida has been derived from a Latin word called hepatica. So it has been derived from Latin known as hepatica, which means liver. Okay, so the reason why they are known as hepatica is that these plants, liverworts, they were used in the treatment of liver, okay, for liver disorders, and this is the reason why they are commonly known as liverworts. Okay, so this class Hepaticopsida is represented by 280 genera. It has about 9,500 species, okay, and they are distributed both in tropical as well as temperate parts of the world. Alright, so let's start with the first point under Hepaticopsida. So as we all know, the main plant body is always gametophyte when it comes to bryophyte. So the gametophyte forms the main plant body, which is independent, meaning that it can synthesize their own food and it is not dependent on any other structures. Okay, the main plant body is dorsiventral, which means that it has a distinct upper and lower surface. Okay, so the upper side that we can see here, in these two structures, this upper surface is known as the dorsal side. Okay, and then if we flip the plant that is facing towards the ground, it is known as ventral side. Okay, so the main plant body will have two sides, the dorsal side and the ventral side. These two surfaces will look different. Okay, they will have different structure, they will have different functions as well. Alright, and then the next point is thalloid. Okay, so the plant body is thallus-like structure. Thallus-like structure, which means that the plant body is not differentiated into root, stem and leaves. Okay, so the plant body has no stem, roots and leaves. Okay, or if they are not thallus-like, they might be foliose-like. Okay, so instead of forming a flat surface, some of the liverworts grow in this manner, okay, in leaf-like structures. So you will have a main axis, this is the main axis, okay, and then you will produce leaf-like structures here, as you can see here. Alright, and the next point is, in the thalloid forms, the plant body is prostrate, which means that the plant will grow flat. Okay, the plant will grow flat on the ground. So if this is the ground surface, the plant will grow flat in this direction. Okay, alright, the plant body will not grow in upright form, so the plant will always grow in prostrate. So the plant is prostrate, it will grow flat on the ground surface. Okay, and then it is dichotomously branched. Okay, so this is the example of a dichotomous branch. This is the example of a dichotomous branch. Okay, see, dichotomous branching means it is a pattern. It is a pattern where the main axis will divide into two equal halves. Okay, and then in the foliose form, the leafy forms, you will have a central axis, and from the central axis you will have 2 or 3 rows of leaves without midrib. Okay, without midrib. So in higher plants, we know that leaves looks like this, right, with a midrib in the middle. But in case of liverworts, midrib will be absent. Okay, so the gametophyte, this gametophyte is the main plant body. It is independent and it is dorsal ventral, meaning it has two surfaces, dorsal side and the ventral side. And then it is thallus-like structure, meaning that the plant body is not differentiated in stem, roots and leaves. Or if it is not thallus form, it might the plant body can also be in foliose form. Okay, like a tiny leafy structure. In the thalloid forms, the plant body is prostrate, it will grow flat on the surface, and it is dichotomously branched. Okay, so from one main axis, 2 equal halves will be produced. Okay, now on the other surface we have the ventral side, right? This is the ventral side. So on the ventral side you will find hair-like structures, right? These hair-like structures that you see here, these are known as rhizoids. So the ventral surface will have many rhizoids which are unicellular, meaning that these are single cell structure. They are unbranched, meaning that they are not branched, and these rhizoids may be of 2 types. This is type 1, this is type 2. Okay, so they may be smooth wall rhizoids or they may be tuberculate rhizoids. So this is how the smooth wall rhizoids look like. And then on some rhizoids you will find peg-like projections here. Okay, peg-like projections, and when you see this type of projections, it means that these are tuberculate rhizoids. Okay, now this is how the rhizoids will look like under the microscope. This is a smooth wall rhizoid and this is a tuberculate rhizoid. And along with rhizoids you will also find the presence of scales. These scales are multicellular. Okay, rhizoids are unicellular and these are multicellular. Okay, they will be present along the rows of the rhizoids, and they have the same function like that of rhizoids. So the rhizoids are known to absorb water and minerals, and they also help to anchor the plant to the substratum. Okay, so if the plant is growing in the soil, the rhizoids will hold on to the soil with the help. The plant will hold on to the soil with the help of rhizoids. If they are known to grow on the rocks or tree barks, they will anchor it to the substratum with the help of rhizoids. The next point is the thallus is either simple, anatomically the thallus is either simple or complex, and it is devoid of mechanical tissues. So this is the thallus of Mertensia. Okay, so if we take a DS section, like this, if we take a DS section and then view the plant body, then we'll see something like this under the microscope. Okay, so what you'll find is that on the dorsal region of the plant, this dorsal region of the plant, you will find that it will have photosynthetic pigments. Okay, you will find photosynthetic filaments here. You will also find the presence of air chambers. You will also find the presence of air pores. Okay, and on the ventral surface, you will find that this region will consist of storage cells. Okay, they will store starch. Alright, and then you will also find the presence of rhizoids and scales. Okay, this type of structure is known as a complex form. This is a complex form. Okay, because here in this plant body you will find that there is a clear distinction of tissues. Okay, clear distinction of tissues between the ventral surface and the between the dorsal and the ventral surface. But in case of Rixia, there is presence of air chamber, there's presence of airborne, but they're not very distinct. Okay, they're not very distinct, and the cells that are present here are also not very distinct like this. Okay, there is no upper photosynthetic region and the lower storage region like that in Mertensia. Okay, so we can also term this as internally homogeneous, and then we can term this complex tissues like internally heterogeneous. Okay. The next point is pyrenoids. Yeah, photosynthetic cells will contain numerous chloroplasts. So the photosynthetic cells that you can see here, these green ones, along with the chloroplast containing cells here, they will contain numerous chloroplasts, but pyrenoids will be absent. Okay, so these pyrenoids are nothing but protein rich bodies So they are just protein rich bodies that are present inside chloroplast, and they help in carbon fixation, and they also help in the storage of starch. So these bionoids are present, they are commonly seen in algae, okay, but they are absent in liverworts, but they are present in Anthoceros, okay, Anthoceros, which is another type of which belongs to the second class, that is Anthocerotopsida. Okay, so pyrenoids are totally absent in case of liverworts. Liverworts are known to reproduce by two methods, vegetative method as well as sexual methods. In case of vegetative method, they will take place through fragmentation, tuber formation, and then through gemma. Okay, so what you'll see is that the plant body, this is known as the apical region. This apical region, the plant will grow in the apical region. Okay, so the direction of the growth of the plant will be in this direction. Okay, so the plant will grow in this manner. So this will be the younger part of the plant, and then this region will be the older part of the plant. So as the plant grows, the older part of the plant body will start to die and decay. Okay, as you can see here, the plant will grow in this direction, and then as the plant grows, the older part will start to die and decay. Okay, and finally all of this will be separated from one another. So each of these fragments will again give rise to a new plant. Alright, and they will also reproduce through tuber formation. So what happens is that during unfavorable conditions, tubers will be formed. Okay, and these tubers they contain starch grains, and then they will grow on the ground. Okay, and then this plant body will die off, and during favorable conditions, these tubers will again give rise to a new plant. Sometimes in Markinsia, there is a formation of gamma cup. So if you look inside this gamma cup, you will find tiny green structures, right? These tiny green structures are known as gemma. Okay, they are known as gemma, and each of these gemma, once it is released from the plant body, they will give rise to a new Markinsia plant. Alright, so they are known to reproduce through vegetative reproduction, through the process of fragmentation, tuber formation, and through gemma. Okay, and the plant body may be monoecious. Okay, so liverworts may be homothallic or monoecious, or they might also be heterothallic, meaning that in monoecious, it means that the plant body will have both the sex organs, male sex organ, and then female sex organ as well. Okay, so the plant body will have both male sex organ as well as the female sex organ embedded in the plant body. Okay, so this is the example of Rixia. As you can see here, archegonium is formed, right? Enteridia is not mentioned here, but enteridia will also be formed inside the plant body. Okay, but in case of Markinsia and other bryophytes, liverworts may be heterothallic as well, meaning that the plant body will produce two. They will have two plant body. Okay, one producing the enteroidiophore and one producing the archegoniophore, and inside the enteroidiophore, the male sex organ will be produced, and inside the archegoniophore, female sex organ will be produced. So liverworts may be homotelic, meaning that the same plant body will produce both the male and the female sex organ. They may be heterotelic as well. So the different plants, the male sex organ and the female sex organ will be born on different plants. Okay. And during fertilization, the male gamete and the female gamete will undergo fertilization to give rise to zygote. Okay, the zygote will start to divide immediately after fertilization. The first division is always transverse, and then this will give rise to embryo, and then the embryo will germinate into a sporophyte. Okay, so the sporophyte may be of two types depending upon different bryophytes. In case of Rixia, the sporophyte that you can see here is a very simple sporophyte. Okay, the sporophyte will consist of only capsule, as you can see here. But in case of Martinsia, the sporophyte will be differentiated into foot, seta, and then capsule. So as you can see, the sporophyte has a foot, this region. It has also a seta, a stalk called seta, and it has capsule where the spores are produced. And each of the spores on germination will give rise to a new plant body. Okay, something like this. Alright, so that's all about Hepaticopsida. Okay, the next class is represented by Anthocerotopsida. Okay, so the class Anthocerotopsida is represented by a small group of 6 genera, and it has around 300 species. Okay, they are widely distributed in both tropical as well as temperate regions. So, okay, the first point is the gametophyte plant body. As you can see here, this is the gametophytic plant body. Okay, this region is the gametophytic plant body, and this region is the sporophyte. Okay, sporophyte, and this is the gametophyte, sporophyte. So the gametophyte plant body is flat, just like liverworts. It is also dorsiventral, just like liverworts. It has a dorsal side and a ventral side, and it is thallus-like. Okay, the thallus is attached to the substratum with the help of smooth wall rhizoids. Now, the difference between Hepaticopsida and Anthocerotopsida is that rhizoids are present in both, but here only smooth wall rhizoids are present. Okay, and then tuberculate rhizoids and scales are totally absent in Anthocerotopsida, whereas in Hepaticopsida, smooth wall, tuberculate as well as scales are present, right? But here only smooth wall rhizoids are present. And then the thallus has a uniform internal organization. Alright, in liverworts, the anatomy, the thallus has both uniform as well as both simple as well as complex forms, right? But in case of Anthocerotopsida, it has a uniform internal organization. Okay, it is not, the plant body is not differentiated into upper photosynthetic region and lower storage region. Here everything is uniform. So the dorsal side will have a upper epidermis, and the ventral side will have a lower epidermis. Okay, and then all the cells are of parenchymatous cells. Alright, and then air pores and air jumpers are totally absent in Anthocerotopsida. And along with that, you'll also find that each of the cells will have many chloroplasts. Okay, the cells will have many chloroplasts along with pyrenoids. Okay, pyrenoids are absent in case of Hepaticopsida, but in case of Anthocerotopsida, pyrenoids are present. So this is a special characteristic feature of in relation to Anthocerotopsida. Okay. And along with that, you'll also find mucilage cavities here in the cells. Okay. On this shaded region, this is the mucilage cavity. So the function of this mucilage cavity is that it holds water. Okay, it holds water, keeping the plant body, the thallus, moist and helping the plant to survive in temporary dry conditions. Okay, so they will hold water and help the plant body to stay moist. Sometimes through this slime pore, and sometimes in the mucilage cavity, algal colonies will also be formed. Okay, so algae like cyanobacteria, for example, nostoc can form colonies. Okay, so what the nostoc will do is that they will fix atmospheric nitrogen, so they will provide nitrogen to the plant, and then the plant in return will provide shelter and moisture to the algal colonies. Okay, so it's more or less like a symbiotic relationship. Okay, and then the sex organs are embedded in the thallus. So Anthocerotopsida may be homothallic as well as heterothallic. Okay, so the sex organs will be present. If it's homothallic, the sex organs will be present. The male and the female sex organs will be present in the same plant, but if it is heterothallic, different plant body will produce different sex organs. Okay, and then finally the sporophyte is differentiated into foot and capsule. So this is the gametophyte plant body, and then after fertilization sporophyte will be produced, right? So this is the sporophyte. So the sporophyte will consist of foot and then capsule. Seta will be absent. And as you can see, this sporophyte body looks like a horn, right? Here it looks like horns, and this is the reason why Anthocerotopsida are also known as hornworts. Okay, so that's all about Anthocerotopsida. Then we'll move on to the next topic. And finally, we have reached Bryopsida. Okay, Bryopsida is the largest group of bryophytes. So it includes about 660 genera, and it has about 14,500 species. Okay, so it is a large number of bryophytes. They are known to occur in all parts of the world, and just like bryophytes, just like liverworts, they are known to grow in moist rocks, dam walls, and also on the barks of trees as well. So the plant body has two growth stages. Okay, in case of liverworts and hornworts, what you'll see is that the sporophyte will produce spores, and then the spores will give rise directly to the gametophyte, right? But in case of Bryopsida, you'll find that the growth phase of this gametophyte has two stages. The first stage is the protonema stage, and the second one is the leafy stage or the gametophore. Okay, so what will happen is that the spores that is produced after spore formation in the sporophyte, here spores that are released will first undergo a protonemal stage. Okay, so what will happen is that the protonema will grow directly from the spore. This protonema is a postrate, okay, which means that it will grow flat on the surface, and it is a branch filamentous structure. Okay, so it will grow postrate, and it has branch branches, right? So it is a branch filament structure, and only through the protonema you start to produce buds. Okay, and through that the gametophore or the leafy stage, you can use any one, the leafy stage or the gametophore will grow from there. Okay, so the gametophyte plant body is known to have two stages: protonemal stage, this, and then through the protonemal stage there will be buds formation, and through the buds this mature gametophore will grow. All right, so this gametophore is more advanced than the gametophyte of liverworts and hornworts, because this gametophore has stem-like structures, and it has also leaf-like structures and root-like structures. Okay, these are not roots, these are not leaves and stem, but they have stem-like structures known as colloids, and you have leaves-like structure known as phalloid, and root-like structures called rhizoids. Okay, so as you can see here, this region is covered by leaves. This is a stem-like structure known as colloid. Okay, and then each of this stem will be covered by these leaf-like structures. These are known as phalloid. Okay, and then you'll find here root-like structures down here. These root-like structures are known as rhizoids. Okay, so the leaves, this is a magnified view of the leaves. The leaves are simple, minute, meaning they are tiny and sessile, which means that petiole is absent. So petiole, in some plants you'll find that the leaves are structured in this manner, right? There's a presence of petiole, but in case of sessile leaves, this petiole is absent. So what you'll see is that this is the stem, and then through the stem the leaves will directly emerge out. Okay, so in case of Bryopsida, the leaves are sessile, so there is absence of petiole. Okay? Petiole is absent with a distinct midrib, which means that there is a presence of a midrib in the middle. Rhizoids, the root-like structures called rhizoids, are branch and multicellular with diagonal cross walls. We've already discussed this. So the rhizoids in liverworts and hornworts are unicellular, but when it comes to the rhizoids of Bryopsida mosses, the rhizoids are multicellular, they are branch, and also there is a special character, that is the presence of cross wall formation in the rhizoids. Okay, now sex organs are situated superficially, forming a cluster at the apex of the stem. So at the apex of the stem here, at the tip, at the top, sex organs are formed. Okay, so the mosses may be both monoecious as well as dioecious. If it is dioecious, it means that the sex organs will be produced on different plants. Okay, so in case of this picture, the male sex organ is produced in this gametophore, and then the female sex organ is produced here. Okay, so as you can see, this is the antheridium is present inside at the tip, and then you also find the presence of archegoniaphore at the tip of the female gametophyte here. Okay, so during fertilization, the sperm will swim towards the egg and then fertilize the egg, and then zygote will be formed, and that through zygote the sporophyte will be produced. Now the sporophyte in Bryopsida may be of two types. Okay, they may be either differentiated into foot and capsule only, or some of the sporophytes of mosses may show all the three characters, that is foot, seta, and capsule. Okay, so that's all about the general characters of Bryopsida. And hey, if you're interested in the books that I use to study and prepare my notes, I've added the links in the description below. Alright, so feel free to check them out, and if you happen to buy through the links, then it helps to support the channel also, okay, with no extra cost to you. So please, if you found this video helpful, then please give it a like. Okay, please give it a like, and then share it with your friends as well. Okay, and then don't forget to subscribe for more content like this. Okay, see you in the next video. Until then, take care and keep learning.